Hello, welcome back to my channel, Bio with Sharuk Shansi. In today's lesson, I want us to cover active transport. Just a reminder, we are still looking at topic number 4 of the Cambridge A syllabus, which is about cell membrane and transport. And the last concept in this topic is active transport. If you're interested in understanding what active transport is, what endocytosis is, what exocytosis is, bulk transport, you're in the right place because that is what we are going to cover. So when it comes to active transport, when you hear the word active, active means we are going to have energy involved. So the process of respiration is going to be very essential for allowing the process of active transport to occur. Why? Respiration is able to provide energy in the form of ATP for the process of active transport. And with active transport, we are going to have molecules, ions, crossing the cell surface membrane. But what makes this kind of transport different is that the molecules are going to be moving from their region of low concentration. Like in this case, we are having two regions separated by this cell surface membrane. We are looking at this ion, which is in green. This side has a low concentration of the ion. You can see the ions are only 3. But within the cell, in the cytoplasm, there is a very high concentration of the ions. So you're going to have ions moving from their region of low concentration to their region of high concentration. But in that case, we are going to have special proteins involved. Which proteins are going to be carrying those ions from their region of low concentration to their region of high concentration. So what happens is that the ion comes, attaches to the protein, and then the protein changes shape and flips the ion into the cell. So that process is by active transport. Why active transport? Because for this carrier protein to change shape, it requires energy from the process of respiration. So what do we note about active transport? Number one, molecules move from their region of low concentration to their region of high concentration across a membrane. And the molecules are going to be carried by special proteins called carrier proteins. However, the carrier proteins do not have a fixed shape. They usually change shape, and that change in shape requires energy in the form of ATP. So that is what active transport is about. It occurs in different forms. Not all the time active transport is going to involve carrier proteins, although in many cases, it will involve carrier proteins. So if we are to define active transport, we can define it as the movement of molecules or ions through transport proteins across a cell membrane against their concentration gradient. What does against their concentration gradient mean? It simply means from a region of low concentration to a region of high concentration. So you find that most of the time in cells, the cytoplasm has a high concentration of ions, such as sodium ions, potassium ions, but the outside of the cell, the tissue fluid, has a low concentration. But the ions keep on entering into the cell, so they will be entering by active transport. Active transport requires energy because the molecules are moving against a concentration gradient from their region of low concentration to their region of high concentration. Active transport requires energy in the form of ATP, and we know that ATP is released during respiration. And one other thing we need to note is that when you look at the carrier protein, it keeps on changing shape. So for it to change shape, it requires energy. That is why this process is referred to as an active process. An example of a carrier protein is what they call a sodium-potassium pump. This is found in the cell surface membrane of our cells. It uses energy to pump 3 sodium ions out of the cell and then 2 potassium ions into the cell. Now, on this protein, it has an enzyme. which breaks down ATP to ADP and an inorganic phosphate so that we have energy released that allows it to keep pumping ions in and out of a cell. So this sodium potassium pump uses energy. It's a special kind of carrier protein. You'll learn more about it as you study coordination in animals in 8.2. But just know it's an example of a carrier protein that uses energy. What other forms of active transport occur now? Sometimes cells need to transport materials in bulk on a larger scale. So they are not going to use osmosis. The cells will not use diffusion. So they are going to use two processes. We have what we call endo. Endo means inside, so inside a cell, and then exo means outside. So we have endocytosis, which is about how cells take in those large molecules into the cell. And then exocytosis, we look at how cells take out large molecules or a larger quantity of molecules outside the cell. Both these processes, endocytosis and exocytosis, require energy. Let's use this diagram to show us what endocytosis and exocytosis are. We can start with exo. Exo, we are going to have the molecules to be transported, placed in vesicles. You learned about vesicles as you studied cells. So this is a vesicle. A vesicle is like a small sac. These are the molecules. You can see there are many, but they need to all be transported out of a cell at the same time. So the vesicle comes, fuses with the cell surface membrane, and then opens and releases whatever it is carrying. In this case, it was carrying a protein. These are protein molecules. So that is exocytosis. Endo is the opposite. We are having the cell now taking up a large molecule to the inside. And in this case, we are looking at an example of maybe a white blood cell taking up bacteria. So the cell forms an enfolding, or it surrounds the bacterium. And then after it encloses it into the vacuole, these two cell surface membranes are going to fuse. Then the vacuole will pinch off and enter into the cell. The vacuole is the same as a vesicle. So it pinches off and enters into the cell. So the cell has taken in this bacterium inside it, and that is endocytosis. So with endocytosis, remember, we are looking at bulk transport. It's bulk because we are having large molecules being carried at the same time, or very many molecules being carried at the same time. So this endocytosis has two types. We could have a cell either taking in liquids, or we could have a cell taking in solids. And that takes us to the first type, which is called phagocytosis, or cell eating. So with phagocytosis, the cell is taking up solids, and we can look at it in this diagram. This is phagocytosis. An example, the cell is taking up this bacterium. So the cell is going to have its cell surface membrane surrounding the bacterium. Then we shall have the cell surface membrane fusing to form a vacuole which pinches off. So here the vacuole has pinched off, and it's containing the bacterium. So here they are showing us the cell has taken up the bacterium by endocytosis, but they are also showing us other processes that happen after the cell has taken up the bacterium. Usually lysosomes come fused with that vacuole. They release digestive enzymes so that the bacterium is killed. And then after that, bacterium has been digested, it has been killed. The products that are not needed by the cell can be released out of the cell by exocytosis. We've looked at exocytosis. So all processes require energy. Endocytosis requires energy, and even exocytosis requires energy. But this kind of endocytosis is particularly phagocytosis because the cell is taking up a solid. Then the other type of endocytosis is pinocytosis, which is called cell drinking. So here the cell is taking up a liquid. And still in this picture, we have our fluid surrounding the cell. The cell needs to take up some of that fluid. So the cell surface membrane surrounds part of the fluid, forms a vacuole, which later breaks off. So this is the vesicle. It has entered into the cell containing the fluid. So usually these vesicles formed by pinocytosis are very small vesicles, but still the process requires energy. So the last one is exocytosis. We are having bulk movement of liquids or solids still to the outside of the cell. And let's use this diagram to show exocytosis. Remember, sometimes a cell manufactures enzymes, it manufactures proteins, but maybe they are needed outside the cell. So they are usually carried in vesicles. We are seeing the vesicle coming from the Golgi body, and it's going to move using energy. Then it will go attached to the cell surface membrane and release the contents outside. Like here, they're giving us an example that in plant cells, those plant cells use exocytosis to release building materials for their cell walls, so that those materials can be used in making the cell wall of the plant cell. So that is exocytosis. Remember that it requires energy. And this exocytosis is so much used in secretion. Like when you look at our digestive system, those enzymes are released by exocytosis. The process is active, it requires energy. We have a question here. They are showing us a transmission electron micrograph of a pancreatic acinar cell secreting proteins. So this cell secretes proteins, but they're telling us there are more mitochondria in this cell than in an average pancreatic acinar cell, and they're asking us why. Remember, we know that mitochondria are so much needed for the process of respiration. So if a cell is active and it's a secretory cell, the process of secretion, the process of exocytosis requires energy. So the cell needs mitochondria so that the mitochondria carry out respiration, providing ATP for the process of exocytosis, for that process of active transport. Thank you so much. So that is the end of active transport. If you like the video, give it a thumbs up. You can share with your friends. Please subscribe to my channel. That is how you show me that you really appreciate my work. So see you on the next one. Bye.